the bipolar, I want you to know these. Bipolar, the retina in your eye has bipolar cells, which are bipolar neurons. Okay, two ends, bipolar. So what do they call them? Bipolar neurons, that's what they call them. So you're going to hear that when we get into vision and the eye. This one over here is bipolar also, maybe not quite as dramatic. And axon here, the dendrites go out here. This one is olfactory cell. What's that mean? Smell. Smell. Olfactory is smell. So these are involved in smell. Where would you look for this? Up in the nose. That's right. They'll be up in the nose, and they're sensory. So these are olfactory sensory neurons. Okay, olfactory sensory neurons. And these are the retinal cells in the eye. You have bipolar neurons, which they call them bipolar neurons, in the retina of the eye. So you got these two that are great examples, but you don't have much else. These two examples are mostly it for the bipolar ones for us to remember for sure. There aren't that many, okay? 99% structurally are multipolar, but 1% are unipolar and bipolar, one of the two. Okay, there's the PEG, which is a neuron cell body. Sorry. Okay. Any questions?
So the unipolars are only in the.
They're neuron cell. Well, those are sensory.
Spinal cord?
Well, or close to it. They're not necessarily spinal cord. Okay. See, they even show it up here, bipolar neuron of retina of the eye. Okay. Here's a sensory neuron. You catch it? You could look at this and never see it. There's a little peg. See the peg? What's it in? It's in something. It's a little bump, isn't it? This is a neuron cell body, right? What's a group of neuron cell bodies? No, well, if it's in the CNS, it is. This is, what was it? Ganglion, good. This is a ganglion, what's plural? Ganglia, and it would be several of these, it wouldn't just be one, okay? So those are some terms to go back and look at. We just went over that, right? No? Okay. Thank you. I was trying to remember if we had just gone over it, I thought we had. Did we see it on the note sheet? There's where it was. So for instance, that was good. Sometimes you can look straight at it and see it.
It's above.
It's the top.
Oh, oh, there it is. Thank you. There are the terms. So we did talk about it. We got down through neuroglia. Here are your structural classifications: multipolar, bipolar, unipolar. Multipolar was the Purkinje neurons in the cerebellum, pyramidal neurons up in the front part of the brain. Bipolar ones were two examples. They're rare, you should know them. In the retina of the eye was one example, and up in the nose, the olfactory sensory neurons were the other ones there that were bipolar. Then unipolar, think of, many are true sensory neurons with their pegs. What are the pegs? Neuron cell bodies. That's true. Sticking out like a peg on a pegboard. Everybody knows what a pegboard is, right? Everybody knows what the rungs on a ladder are. I have to always check. Okay. I didn't know if pegboards were still in fashion or not. I thought they were. You could have that in your garage and you hang your tools on it, stuff like that, pegboards. Okay. Well, you could do the structural idea. Let's talk about the structure of the neurons and classify them that way. 99% are multipolar, the 1% are the mixture of bipolar and unipolar. But what about function? Everybody knows that nerves can be sensory or they can be motor, and we get all tied up with that and say, oh, wow, wow, wow. But the thing is, 99% are not. They're not sensory, they're not motor, they're not true sensory, true motor, they're interneurons. So I've not heard of that, now you have. Interneurons do the processing. So up in your brain, the surface of the brain is all interneurons, and you're doing a lot of thinking there, okay? So 99% functionally are interneurons, and that's the cerebral cortex up there. We don't have a cerebral medulla. We do have a cerebral cortex though. And it's only about 1/8 of an inch thick, so it's not real thick, but it's all twisted, okay? So once again, 99% of neurons functionally are interneurons, whereas 1% are either true sensory neurons or true motor neurons. What makes them true sensory? They're bringing information back to the brain and spinal cord. What makes them true motor? They're taking commands out to muscles, things like that. So could there be neurons carrying information from these? Yes. That aren't really true sensory neurons? Yes. Or not true motor neurons? Yes. Okay, so we get into that. Here's a cute little thing. There you go. A little voltmeter. What does it measure? Voltage, of course. And those units come in millivolts. Millivolts. We don't need big volts. Millivolts are good. That's 1,000th of a volt. That's enough. Okay. You stick this cute little machine on an axon. You put this part inside the axon. This part's on the outside. You can check out the difference between the charges on the outside versus the charges on the inside, based on the inside more. So it's negative inside compared to the outside. So the voltmeter says negative 70 millivolts. And that's pretty good for the voltmeter here. Okay, y'all got that?
Is it necessarily is it the inside of the cell in general, or the
It's inside of the axon. I'm not sure 100% that it stays the same the whole way through on the membrane. Probably does, but I won't say for sure. Okay? Now, I've got a question for you. What if you had a channel here, a sodium ion channel? Okay? Where do you have more sodium ions? Outside. So if you had a sodium ion channel here that was opened up, which way would the ions go? Inside, because this is negative in here, and they're positive. Okay? What would that do to the inside?
lower the voltage?
It would make it go towards positive.
Yeah.
Okay? So it go towards positive. Right here, it's pretty polarized, isn't it? Positives on the outside, negatives on the inside. But if you're leaving, if you're coming in with a positive charge, what would you say? You were polarized before, you're doing what if you're coming in with positives? What?
Depolarizing?
I would go as far as to say depolarizing.
Yeah, reducing the negative.
Yeah, you're depolarizing, okay? So we do talk about depolarizing, and remember at neuromuscular junction, the positives coming in, we're depolarizing it, okay? So, we're going on here, and I skip over some of the stuff because we don't need it, and we're gonna skip over it. But I wanted to go to this next. Everybody recognizes this is one action potential. And we see the setup, little voltmeter here on the axon. And what I want you to think about, because you may not have this down real well, an action potential travels along the axon out to the glucon. An action potential, what is it? It's a change in voltage. So you have a change in voltage that's progressing along here. And this is one spot, but each spot will go through this process. Like a bunch of dominoes, you'll fall down through here, okay? So if it was negative 70 millivolts, and that was your resting state, suddenly you start changing because sodium ions come in, for instance, and you start depolarizing. But you can get up to a 0 here. So the inside and the outside at that point are equal, and you can go up to positive 30 as part of your action potential. and then the sodium ions stop coming in, and now who comes out? Potassium ions, okay? They flow out of the cell, and that's called repolarization. Repolarization. So they clean up the mess. The sodium ions make the mess depolarizing, and then the potassium ions are the ones that are supposed to fix it back the way it was. Remember your pink neighbors and your purple neighbors? Yes? Okay. Well, the pink ones are depolarizing with sodium ions, and the purple ones are cleaning up the mess afterwards by repolarizing with potassium ions. Any questions? So, look, we're repolarizing. How far we go down? Well, we went through threshold, and I didn't mention that. But as you're depolarizing, once you hit threshold, once you get to negative 55 millivolts, that says, I'm sorry, you may want to, but you can't stop the action potential. It happens. Okay? So it happens. But as you're repolarizing, you get down here to negative 7, and it keeps on going beyond negative 70, down to negative 90. And you say, oh, what have we just done? We didn't just repolarize, we hyperpolarize. And then it comes back up and stays at negative 70. What would keep it holding there at negative 70? Which one?
I said feet, I don't think that's it.
Yeah, that was nice, but I don't think it was right. Okay. How about the sodium ion, potassium ion pumps? Sodium ion, potassium ion pumps will keep it at negative 70 afterwards. See, it was negative 70, it's going to be negative 70 again, and this was just a weird thing that happened, the action potential. But if it happened to the one spot, it's going to happen to the next one, and the next one, and the next one, in a progression. So, action potentials are voltage changes in a progression down here. Okay. And you do go to negative 90 approximately, until you come back up. It's like, oh, it was so much fun repolarizing that we just couldn't stop, and we went a little too far, but we'll come back up. Any questions? Let's take a look at our pink neighbors and purple neighbors, okay? The pink ones are the sodium ion channels. Give me the full name.
Chemically dangerous.
No little green dots here.
Voltage.
Voltage gated sodium ion
ion
channels. How do you know they're voltage gated? They're the foot pedals. See them? Foot pedals. Okay. So sodium ions want to come in. There are some in here, but they really mostly want to come in. So they can do that. Every one of them is positive, so they're changing the inside to more and more positive. This is something you don't have on the purple one. It's called an inactivation gate. I like to think of it as a plug on a bathtub. You got water in the bathtub. You want to empty it so you go get a sandwich while it's draining, then you'll come back and clean it, and you come back and the stupid plug got in here and blocked it. So what happened? It did not drain. Okay? This is called an inactivation gate. It can block this. So the foot pedals could be up, but you still can't get through because of this. Only that one does it. The purple one doesn't have an inactivation gate. And is it a big deal? No. But I mentioned it to you. Okay? If the pedals are down, it's closed. But if the pedals are up and it's a sodium ion channel, it could still be blocked. So we've got the sodium ions coming in. Well, not yet. We know they're coming in if the pedals are up. So it's coming in. And here, it got blocked, but the potassium ones are going out, which they wanted to do. And if these are positives leaving, what's that going to do to the inside? It's going to make it more and more negative again. So we're getting back down to negative on the inside, positive on the outside. Okay? So you want to look at repolarization, hyperpolarization, depolarization. Just be sure with your study group you've got that figured out. sort of looks like the chicken that ate New York City, okay? These are actually axons, axons. This is the nucleus, the neuron cell body actually. So there we go, that looks familiar again, doesn't it? And what's this thing coming in? It's an axon coming into the soma, the neuron cell body, okay? Here's an axon coming to a dendrite. Here's one coming to another axon, okay? So here's a synapse that's an axosomatic, axodendritic, axoaxonal. And we see jelly rolls, so this must be PNS, right, now you got it, okay? I think I wanted to mention it. Yeah. What happened with Granny and her little VW convertible? She goes zap, 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 and she got all this power and could lift it off her grandson, right? What do we call that? Zap, 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 zap.
Fiber muscle recruitment.
Recruitment would be having a number of motor units. This is not so much that.
Summation.
What?
Wave summation.
It's a summation. Which one was it?
Wave summation.
Wave summation. That's another name for what?
Temporal summation.
Temporal summation. So you hit one place multiple times, and you get more power if you don't let it get completely relaxed. So that was our best shot at it. And you can actually get to tetanus if you keep working at it. But over here, we said, we're not doing that. We're gonna zap here, zap there, zap everywhere. Well, not really, but different places. That's not wave summation, that's not temporal summation, that's spatial summation. So if you're stimulating at several different places at the same time, has to be at the same time, then it's spatial summation. See, this was the same spot, but it was over and over again. Any questions?
And that's,
This was wave summation equals temporal summation. This was spatial.
so you can have a cell body or a neuron cell body that like a bunch of neurons attached to?
Yes.
Instead of just one repeating?
In all honesty, in natural life, there are loads of connections like this, but they're not touching. They have the synapse, but they're real close, but not touching.
Does it ever do both, like a bunch of them attached and the summation or the temporal?
Oh, these were not attached.
Well, right, but, you know, maybe.
Even, I think you could have both.
Oh.
I don't see why you couldn't. You can have both. Granny had a lot of help there getting that car off, okay?
You know what? I'll tell you. I don't know this. I'll tell you the picture.
So look at this, if you want to see a lot of them, there they are. You can have a lot of them doing their thing. One neuron talking to other neurons.
I think I saw a video once, don't those, I wanna say connections, but like how all the axons are coming to a body, like
Yeah.
they're dynamic, they move and make new connections and disconnect.
They possibly can. It's not like they're real mobile. But again, this is not tacked down because they're not attached.
Right.
So I think it could kind of float over here, float over there. type thing. Okay? Why red and green? They're not celebrating the season. Why red and green? Well, let's go on to the next page, and I think it will become clear. Different results.
It's like half written, yes.
It looks a little that way, it's not 100% that way. Here is a neuron with an axon coming to it, not touching, but very close, synapse, and it zaps, but it's not enough to get an action potential. And then it zaps again, and it still didn't get it. To get an action potential, you had to be over a threshold, over -55, and it didn't quite make it 2 times. But then it says, like, we're gonna zap, zap, zap type thing. And that was enough. If you didn't let it relax enough, you zap the second time, you get an action potential. So this is temporal summation. Here we didn't get any summation. Down here, you had the two axons, not one, but two, different places. And if they both go at the same time, then you have spatial summation. And that was enough to put us up over threshold, we had an action potential. You cross threshold, you're gonna have the action potential. Here, we have one green one, and when you do this, you're gonna make what's called an EPSP. EPSP, a graded response. EPSP. It's called excitatory postsynaptic potential. Excitatory postsynaptic potential. But is it possible to have an inhibitory one? Yes. An IPSP, inhibitory postsynaptic potential, which is going to try to keep you from having an actual potential. So how are you going to know what happens? Well, you have to take both of them into consideration and see who wins out. So the red ones are inhibitory, the green ones are excitatory. EPSP. Any questions?
Why would one person inhibit?
Why would one inhibit and one excite?
Yeah.
That's a good question. It may be the kind of axon we got here and the kind of neurotransmitter that's being moved. Because, as you're going to find out today, there are not just two.
Okay.
If you want to memorize all, it will be over 50. Yeah. And so there are different neurotransmitters, and it's surprising what they are too, which we'll look at in just a little bit. Okay? So you got it, no summation, temporal summation, spatial summation, and you can get both spatial and temporal. You can get EPSPs and IPSPs. EPSPs excitatory, IPSPs inhibitory. And you gotta figure it out. Who has to figure this out?
The neuron.
The
Oh, the.
axon hillock, right? It decides from all the information and either sends an action potential or not. Yes.
Could the EPSP and the IPSP be ways of protecting the body?
They can be used for different things. All of this has potential, you know, having a lot of synapses, having just a few, all of it matters. And whether it's inhibitory or excitatory certainly matters. So the brain and the body is pretty complex. We're looking at it from a simplified view, but it could.
Because I'm thinking EPSP could be like, you're lifting a box and it's heavier than you think it is, so you send more impulses to try to,
To make up for that.
Yeah.
Yeah.
and then IPSP is to tell you to stop.
It also could be like, you realize it's heavier than you should be doing, you're afraid of damaging your back, so you drop the box. I mean, something like that could happen. So there's interactions, there's based upon things going on there. So this just gives you more connections and more possibilities. And like I showed you back up here, that's quite a few connections. And actually, it's probably a small number. I think it's much more than this. The connections are amazing how many there are. And they're not touching, they're real close, but they're not touching. Well, here it comes. 50 or more neurotransmitters